Good afternoon, friends. Now we have already taken a series of lectures for UV-Visible Spectroscopy related to Analytical Chemistry chapter number 5 of 2019 pattern. Now in previous lecture we have already learned about the applications associated with the colorimetry and spectrophotometry. Now in this lecture we will learn some part of the spectrophotometer. Now there are two types of the spectrophotometer. One is single beam spectrophotometer, another is double beam spectrophotometer. Now the name itself suggests that single beam spectrophotometer involves the single beam only. Now here, typical diagram of single beam spectrophotometer is as shown in figure on this world page. There are the two sources employed in the spectrophotometer, that is single beam. One is UV source of light, another is visible source of light. We know that for visible source of light, tungsten filament lamp generally used, while for UV light source, we use the discharge lamps like mercury, xenon, or we can say any type of deuterium discharge lamps. Now when this source of light strikes the surface of mirror, a collimated and parallel beam of light is obtained. This parallel beam of light is focused on the monochromator, where monochromatic radiations are obtained, and these monochromatic radiations are further passed to the reference cell and sample cell. There are two types of the cells used. Now one is reference cell, another is sample cell. Now here through the reference cell, intensity of transmitted radiations are obtained, while for the sample cell intensity of transmitted light is obtained, while through the sample cell intensity of transmitted light is obtained, while for the reference cell intensity of incident light is obtained. Both are recognized to the direction of detector. Now here some theory is associated with the typical diagram is given below. It consists of two light source, as I have already said. It consists of two light source, tungsten halogen lamp for visible and deuterium lamp for far ultraviolet visible region. They are mounted on a movable arm for correct operating position. Now these two light sources are put up in such a way that they are mounted on a movable arm for a correct operating position. There are also two phototubes detectors. Now there are two photo detectors are employed. One is for the photo reference cell, another is for the sample cell. There are also two phototube detectors are employed. Out of one is brought to the focal point of the lamp. Now focal point is nothing but the corrected or converged point or center of gravity point. The light enters into the instrument through an entrance slit and is collimated and focused on a dispersion element like diffraction grating or prism. Now the light is obtained from the source, and this light source is focused on springed on the entrance slit, or it is bombarded on the entrance slit. Through the entrance slit, a parallel beam of radiation is obtained. This parallel beam of radiation is nothing but the collimated lens or collimated beam of light. And this collimated beam of light is focused on the dispersion element. Now what are the dispersion elements are employed? One is there diffraction grating or filter, diffraction grating or prism. Now both, or you can say out of one, acts as a monochromator. So here this diffraction grating or prism acts as a monochromator to obtain the monochromatic light from the parallel beam of light. The light of desired wavelength is selected by rotating monochromator which impinged on sample. Now we get a proper or appropriate or desired wavelength through the monochromator. So monochromator, why it is so used? To obtain the selected beam of radiation, selected wavelength of radiation. Monochromator is used, and the light transmitted from the monochromator is now passed towards the sample. And whatever the intensity of transmitted radiation through the sample is measured and recorded as a absorbance or percentage transmittance. The intensity of transmitted light through the sample is measured and recorded as absorbance or percentage transmittance. So we will get the output in terms of absorbance or percentage transmittance through the photocell detector. With this content, I conclude this lecture. Thank you very much for patiently listening.